Trematodes, commonly known as flukes, are flat, leaf-shaped organisms. They are hermaphroditic, containing both male and female reproductive parts, except for the schistosomes, which are unique because they have separate sexes. Trematodes always require an intermediate host, and the first intermediate host is always a snail. Their eggs are operculated or capped, with the exception of the schistosomes. Fasciolopsis buski, the giant intestinal fluke, infects humans when they ingest raw aquatic vegetation such as water chestnuts that contain encysted metacercaria. The diagnostic stage is the presence of characteristic large eggs in feces with a small operculum, which resemble those of Fasciola hepatica. Clinically, infection causes diarrhea and epigastric pain, and the severity of symptoms relates directly to the number of worms. This parasite is found in Asia. Fasciola hepatica, the sheep liver fluke, is also acquired by eating raw aquatic vegetation containing encysted metacercaria. Its eggs, found in feces, resemble those of Fasciolopsis buski. Disease results when migrating larvae trigger an inflammatory response in the liver, leading to obstructive jaundice, fever, vomiting, diarrhea, and eosinophilia. Sheep and cattle act as reservoir hosts, making this fluke common in sheep-raising regions. Clonorchis sinensis, the Chinese liver fluke, infects humans who eat raw fish containing metacercaria. Its eggs are operculated with distinctive shoulders and a small comma-shaped appendage at the abopercular end. Found in East Asia, including China, Korea, Japan, Taiwan, and Vietnam, it has been associated with bile duct neoplasms. Paragonimus westermani, the oriental lung fluke, is acquired by eating raw crustaceans such as crabs or crawfish infected with metacercaria. Diagnosis is made by detecting eggs in feces or sputum, sometimes appearing as reddish-brown flecks in the sputum that resemble iron filings. Eggs are operculated with shoulders and a thick abopercular shell. Clinically, light infections may be asymptomatic, while heavier infections lead to chronic cough, mucus production, and marked eosinophilia. This parasite is common in parts of East and Southeast Asia, including Japan, Korea, China, and the Philippines. The schistosomes, also known as blood flukes, are the most medically important trematodes because of the severity of disease they cause. Unlike other flukes, they are not hermaphroditic. They exist as separate male and female worms. Adult flukes live in venules, with Schistosoma japonicum and Schistosoma mansoni inhabiting mesenteric venules, while Schistosoma hematobium lives in bladder venules. These worms can survive in the human host for 4 to 35 years. The snail is the intermediate host, and humans are infected when cercariae in contaminated water directly penetrate the skin. Diagnosis is made by finding characteristic eggs in urine or feces. Schistosome eggs are differentiated by their spines. S. mansoni has a conspicuous lateral spine, S. japonicum has a small lateral knob, and S. hematobium has a terminal spine. Clinically, schistosomiasis causes progressive chronic inflammation of the liver, intestines, and bladder. A dermatitis known as swimmer's itch may occur when cercariae from non-human hosts penetrate human skin. Eosinophilia is common. Specific symptoms include hematuria with S. hematobium infection and bloody diarrhea with S. mansoni or S. japonicum. Schistosomiasis occurs in many regions, but in the United States, transmission does not occur due to the absence of suitable snail hosts. When looking for parasites, remember that not all are found in stool specimens. Some are detected in other body sites. In the blood, you may find microfilaria larvae, except for Onchocerca, which is instead found in the skin. In the sputum, eggs of Paragonimus westermani, adult Ascaris lumbricoides, and various forms of Strongyloides stercoralis, whether adults, larvae, or eggs, may be seen. In liver or bile fluid, you can detect Echinococcus granulosus larvae, adult Clonorchis sinensis, and adult Fasciola hepatica. In the urine, Enterobius vermicularis eggs and Schistosoma hematobium eggs can be found. Finally, in the mussels, Trichinella spiralis larvae are diagnostic. So, beyond stool exams, always consider blood, sputum, liver fluid, urine, and muscle tissue for parasite detection.